And our last speaker of the session is Chandler Sutherland from Berkeley, California, who will be talking about plant immune responses.
Yes, thank you to the organizers for giving me the opportunity to represent Kingdom Planta here in our non-human genomics session. I'm looking forward to tell you about recent work from myself in the Cross-Oleva lab at UC Berkeley, studying how these really rapidly evolving immune receptors in plants generate really rapid genetic diversity, specifically the contributions of mutation and selection to this process. And I hope to convince you by the end of the talk that innate immune systems are a really fascinating case in which to study the evolution of mutation and mutation rate. And that's because the innate immune system and immune systems in general require very high genetic diversity. On their face, mutations are deleterious, and there's been a lot of really elegant evolution of DNA repair machinery to keep mutation rates low across the genome. But the immune system is a case where high genetic diversity is required to keep pace with rapidly evolving pathogens and disease. And that's allowed for a lot of really interesting, and if we look at them retrospectively, risky evolutionary strategies across the tree of life. Maybe most familiar to us is the clonal selection-based adaptive immune systems in vertebrates, which is a self-mutagenic process. There's somatic hypermutations and rearrangements that allow for the really stunning diversity of our antibody receptors that allow us to respond to new diseases on the timescale of a few days to a week. On the other end of the tree of life is template targeting-based adaptive immunity, or more familiar to us as CRISPR, which includes incorporation of exogenous DNA into the host genomes. Again, pretty evolutionarily risky, but very successful for these bacteria that deploy this type of immune system. Other work across the tree of life, specifically in mollusks and other arthropods, have seen this really high somatic diversity in immune receptors without a corresponding mechanism of selection. And so without an exposure to a pathogen and without any type of initiation from what we can tell, there's really high diversity at immune receptors in these mollusks in this system that we've termed anticipatory immunity. But my question and the question of my lab is what's going on in plants? And other people in the lab are working on fungi. Because plants have this innate immune system, and we know that innate immune systems require this high genetic diversity. So what systems are maintaining and generating diversity in these plant immune receptors? And do they fall under this anticipatory immune framework? And so the part of the plant immune system I'll be discussing today is the NLR-based intracellular immune system. And so NLR stands for nucleotide-binding leucine-rich repeat receptors. And they are this modular intracellular protein that recognize pathogen-secreted proteins called effectors. And there's two main modes of action for these NLRs. They either directly bind to these pathogen-secreted effectors, or they indirectly recognize infection by binding to plant proteins that are often targets of the effectors, so monitoring for this infection. And I want to highlight these two modes of action because you can imagine they're under very different selective pressures. And so these direct binders are in very intimate co-binding relationships with these pathogen-secreted effectors and are often under positive diversifying selection to maintain those relationships. Whereas the indirect binders are in more stable partnerships with the plant proteins and are therefore usually more conserved. And I want to take a minute to appreciate the fact that these are all germline-encoded in plants. And so we have direct binders that are encoded directly from the DNA that persist over the organism's lifespan. And so when we think about plant innate immunity, we have appreciated very high diversity in these immune receptors at a population level. As we enter into the pangenomic era of plants, these NLRs are among the most diverse proteins across the genome. And when we think about amino acid diversity, it's a combination of both the genomic processes that generate variation and selection that promotes its maintenance. You need mutations to be able to have variation, and then you need selection to keep those mutations to being lost by drift. And I think of this as kind of a forensics experiment. The data that I have, is an alignment of amino acids, but we have to kind of go back and try to differentiate the relative contributions of these two processes to get to this question of where variation is coming from and how it's maintained. And so we needed a metric that was kind of agnostic to signatures of selection and population structure. And so we ended on Shannon entropy, calculated along each amino acid position in an orthogroup alignment of these NLR immune receptors. And this was work done in collaboration with Daniel Prigozhin at LBNL. And here I'm plotting entropy at the 10th highest position of an alignment versus the number of NLRs. And we chose this 10th position to capture these proteins that have multiple amino acid sites with very high diversity. And this is our distribution in Arabidopsis, the model plant. And as you can see, it's a bimodal distribution. And so this allowed us to kind of split the bimodal distribution into two gene sets, which we're calling non-highly variable NLRs and highly variable NLRs. This is the last acronym I'll give you to remember, so keep it with you. But if you think back to how these NLRs work, all of our known direct binders were in this highly variable category, and all of our known indirect binders were in this non-highly variable category. And that's not to say that all highly variable NLRs are direct or vice versa, but it was a good sanity check that this metric of diversity was capturing the information we were looking for. It's this kind of unbiased metric of amino acid diversity at the pangenome intraspecies level. And so with these gene sets, we can begin to compare highly variable to non-highly variable NLRs, but also compare NLRs to the rest of the genome. And so we started with a few genomic features. And we found that highly variable NLRs are more expressed than non-highly variable NLRs. I'm showing you rosette leaf tissue here, but we found it to be true across the vast majority of tissues, including really important reproductive tissues like embryos and seeds. And when we compare this to the rest of the genome, this x-axis, I've ranked all of the genes in the genome by expression, and then we're looking to see where these highly variable and non-highly variable NLRs fall in this ranking. And we can use gene set enrichment analysis to see if there's any statistically significant enrichment. And we found that highly variable NLRs are among the most expressed genes in the genome. This is no pathogen exposed to these tissues. These are just standard greenhouse conditions, whereas non-highly variable NLRs look like the other genes in the genome. So we see this high expression as kind of a genomic feature associated with highly variable NLRs. We did the same analysis for methylation and found that non-highly variable NLRs are more cytosine CG context methylated than non-highly variable NLRs. And in Arabidopsis, CG methylation, while it does confer an increased likelihood of mutation by spontaneous deamination, it's often associated with very stable housekeeping genes. And so there's been postulation that by some unknown mechanism, this CG methylation confers stability to these genes. And when we look across the genome, it's definitely not as strong of a signal, but we do have significant enrichment of our highly variable NLRs among the most hypomethylated genes in the genome. And finally, we looked at distance to transposable elements. And we found that the highly variable NLRs are much closer to TEs than non-highly variable NLRs. And this is actually a median TE distance of 0. So these highly variable NLRs often have transposable elements in their introns or 5' or 3' UTR sequences. And the enrichment is quite striking. And so the highly variable NLRs are among the closest genes to transposable elements in Arabidopsis, whereas non-highly variable NLRs look like the rest of the genome. They're very distributed across. And so with these features, we wanted to see what signatures of selection, if any, differentiate these two groups. And so we started with Tajima's D and nucleotide diversity, which is represented by this pie. And to do this, we first had to look to see what the distribution was across all of Arabidopsis. And as been described previously, the Tajima's D runs a little low in Arabidopsis due to some historic bottlenecks. And then we have kind of mostly genes have very low nucleotide diversity with a bit of a tail. And if we overlay our NLRs, we observe primarily high nucleotide diversity of highly variable NLRs. Highly variable NLRs are defined by amino acid diversity, so this was what we expected. And non-highly variable NLRs mostly look like the rest of the genome. In terms of Tajima's D, our highly variable NLRs are actually not significantly enriched in either tail. And so we can't say from this that highly variable NLRs are maintained by strong balancing selection. Non-highly variable NLRs, however, are significantly enriched in the bottom part of this tail, indicating that purifying selection may be helping to keep their sequence conservation. And then we did some branch site statistic models looking at positive selection and did see that highly variable NLRs have more codons under both episodic and pervasive positive selection. And so we wanted to dive a little deeper into nucleotide diversity and the contribution of mutation. And so here we looked at both synonymous nucleotide diversity and non-synonymous nucleotide diversity. And in both cases, highly variable NLRs have an increase in this non-synonymous and synonymous diversity. But when we look at their ratio, which is kind of a rough metric for positive selection at the gene-wide level, we see no difference. And so one explanation for having a higher non-synonymous and a higher synonymous nucleotide diversity with no difference in their ratio is an increase in mutation. And this was corroborated by mutational likelihood based on chromatin state, which we calculated in collaboration with Dr. Gray Monroe at UC Davis. And we observed that highly variable NLRs have a 30% higher mutational likelihood based on their chromatin state. And so if we zoom out and think about what's going on in Arabidopsis, we observe this higher mutational probability and distinct genomic features that by some unknown mechanism are likely increasing this mutational probability or affecting it. And we observe positive diversifying selection on highly variable NLRs and purifying selection on non-highly variable NLRs. But this is all in Arabidopsis, and we wanted to see what these features would look like in a different species, namely a crop. And so we looked to maize, which is a model organism, and has really wonderful genomic resources to begin to investigate these questions in a different system. And first, we did our highly variable NLR analysis and found that there is a depletion of total NLRs and highly variable NLRs in maize, likely due to domestication-related bottlenecks. But we do observe these collapse down to 4 distinct highly variable NLR clades in maize. So despite domestication decreasing a lot of our genetic diversity, there were still 4 clades that we could begin to examine with highly variable character. And so with maize, we were able to look at pangenome and across tissue types of expression. And so here I'm showing each column is an NLR clade, and I'm showing the percent of the clade in these binned expression states. And many NLRs are silent. Some of these are likely pathogen-inducible. But as we move through the non-highly variable clades, some are tissue-specific and some show constitutive expression. And then if we zoom into just our highly variable NLR clades, we show distinct expression states within the same orthogroup or within the same clade. Particularly if you look at these RPPM-like and RPPC-like clades, these are all orthologs. Like this is as close to an allelic series as we can get with this data. And we see some that are constitutively expressed across all tissues, some that show tissue-specific expression, and some that are silent. So we're seeing this really rapid differentiation of expression states correlating with our sequence diversity. And we also could look at methylation. And so in maize, most genes exist along this continuum of CG methylation here. And similar to Arabidopsis, high CG methylation is associated with sequence conservation, housekeeping type genes. And then we have this other metric of CHG methylation. And if you have both and you end up in this cluster up here, that's associated with TE-like methylation. So you're silenced, and there's often a lot of sequence divergence in these genes. So I'm showing you 4 representative lines, and this is all of the genes in the genome plotted. And if we overlay the NLRs, we can see that there's variation in both non-highly variable NLRs and highly variable NLRs in their methylation states. Most NLRs exist along this continuum of CG methylation, but in a few lines, you'll get these NLR clusters in this TE-like methylation state, and some that are in this ambiguous state. And so when we think about the immune system, as we've seen in maize, we observe a reduced but present highly variable NLR clades despite these domestication bottlenecks in a really important agricultural crop, and also expression and methylation variation associated with this sequence variation. And so I want to bring it back to our tree of life and argue that the plant immune system evolution fits this anticipatory model at a population level. We have this subgroup of NLRs that are evolving quite rapidly. They have distinct genomic features and signatures of selection that are helping to promote this diversity and a higher likelihood of mutation. And so with that, I'd like to thank my lab and my collaborators on this project, Dr. Prigozhin, Sanjay, and Dr. Gray Monroe, the JXTX Foundation for funding my attendance at this conference, and my PI, Ksenia. And with that, I'd be happy to take any questions.
Great, thank you.
Really nice talk. Did you happen to check to see with any of the NLRs whether they were sharing promoter? They had bidirectional promoters, or any selection was associated with that?
I have not looked to see if there's any promoter sharing happening at the NLRs. One interesting thing that came out of the Arabidopsis work was, NLRs are often in these clusters, but it seems as though highly variable status does not require a cluster position. So we do see these NLRs that are singletons and would obviously have a discrete promoter that maintain highly variable status across the genome.
Thank you.
Hi, nice talk. I had two questions. One is about, have you looked at wild maize and whether it has more highly variable NLRs? And the second question I had was how plants also have another immunity pathway using RNAi, and how there might be correlations in the diversity of RNAi pathways and diversity of NLRs.
Yes, I haven't looked at wild maize, but I would expect that in these teosinte populations, you would see higher diversity of NLRs just due to the kind of current framework about how domestication impacts genetic diversity, specifically in immune receptors. So likely there would be more highly variable NLRs, but I have not checked. And then in terms of the RNAi question, I think that there's also other types of plant immune receptors, and we focus primarily on amino acid sequence here to capture some of these broader trends. But from what I've started to look into, the RNA level, there's a lot of diversity also. And even at the alternative splicing level, we observe high differences in alternative splicing in NLRs, but we haven't quantified that difference yet. But there's lots of layers in which you could achieve diversity beyond just amino acid or nucleotide sequence level. Thank you.
In the back there.
Thank you. This is a fantastic talk. Can you tell me a little bit about any findings you may have had with membrane-bound, leucine-rich repeat receptor-like kinases?
Yes, so the Arabidopsis work is based on the NLR-OME, and so it was an enrichment-based sequencing approach specific to NLRs. And so we couldn't look at NLRs in Arabidopsis until recently. There's lots of long-read pangenomes that have come out, and so we're actively doing that type of work. And it's likely that the membrane-bound receptors, we will see highly variable character in some of them. And then in maize, the leucine-rich repeat membrane-bound receptors did show up in this kind of highly variable analysis in the whole genome scale. So far we've only done the feature analysis in NLRs, but we are seeing similar highly variable character in those RLPs and RLKs.
All right. Thank you again, Chandler. And let's thank all the speakers in this morning's session.